My name is Ariel Ben-Ezra. I work at the Mount Sinai School of Medicine in the psychiatry department under the leadership of Dr. Jose Moreno. And we studied the role and structure of a serotonin 5A glutamate 2 receptor in our mouse model of schizophrenia. Schizophrenia is a disease that affects 1% of the population. It's characterized by positive symptoms, negative symptoms, and cognitive impairment. Positive symptoms being hallucinations and delusions. Negative symptoms would be social withdrawal, anxiety, depression. And cognitive impairment would be behavioral issues, memory loss. And glutamate and serotonin have been implicated in the schizophrenia disease. So we took a look at two of those receptors in the cells in the brain. So this is the complex, this is the glutamate 2, and this is the serotonin 5A, and this is the cell membrane. And basically, we wanted to find out how these two interact. So our first experiment was in mice. We had our stress model of schizophrenia, so we induced schizophrenia in mice. And we used radioactive tags to quantify the different proteins and saw that with the serotonin in the schizophrenic mice, there was an increase in serotonin compared to the control. And in the glutamate 2, we saw there was a decrease in the level of protein than in the control. And that just showed the imbalance in the protein levels. So then we wanted to look more at this complex and see how it interacted. So we focused on the TM4 strand of the glutamate 2. We just kind of chose one to focus on. So we looked at this TM4 segment, this one right here. So we got the sequence of the amino acids and we used glutamate 3, the sister protein of glutamate 2, which has no relation to the complex, but it was close enough in its sequence and had certain changes that we wanted to see why glutamate 2 is used in this complex and glutamate 3 has nothing to do with the complex. So these would be the new regions, the new amino acid sequence. So we used the glutamate 3 and replace these little segments. This is farthest from the inside of the cell, and this is closest to the inside of the cell. And so we used these chimeras to basically just run our dimer test. So we used we excited our proteins and were able to detect how much dimer was created, how much of this complex was created by each of these proteins. So we saw with the glutamate 3, we expected there to be no protein no complex created, and there was no complex created. And with the glutamate 2, there was a complex created. But when we replaced the region furthest from the cell in the glutamate 2, when we replaced it with the glutamate 3 in the region furthest from the cell, we saw that there was no complex created, whereas when we replaced the region closest to the cell, there was dimer created. So basically, we found that these 3 amino acids, the ones that we replaced with the glutamate 3, were absolutely necessary to create this dimer, because without them, when we replaced them, the dimer wasn't created. Finding this was important because in future, when we hopefully wish to develop a medicine, disrupting this complex could be important, and since we know these are necessary, we could try to find a medicine that could disrupt those. Our last experiment was to relate the complex back to schizophrenia. So these were done in vitro, which is in cell culture. So we took a group of cells that had both serotonin 5A and glutamate 2, and then we took a group of cells that only had serotonin, and we added hallucinogenic drugs like LSD and DOI. and added different concentrations. And basically, since these hallucinogenic drugs only bind to serotonin, it should be an equal level of the hallucinogenic binding across the board, but we didn't see that. We saw that in the presence of this dimer, there was an increased level of hallucinogenic binding than in the only serotonin cells, which shows that that these hallucinogenic drugs were in some way attracted to the dimer rather than only serotonin. Okay.